Good afternoon, everyone. It gives me great pleasure to chair our session on research and clinical trials. My name is David Parr, and I'm a respiratory physician in Coventry, UK. I've been involved in Alpha-1 as a researcher and clinician for a long time, nearly a quarter of a century, which makes me feel very old. I won't say any more about myself because there's a detailed biography on the website, and I'll also be speaking in the next session. We have three superb speakers for this session, all of whom I know very well. It is a very active time for research and trials at the moment, and I'm sure that you will find their presentations extremely interesting and informative. I anticipate there will be a lot of questions in this session and would like to remind you that the next session will address the subjects of disease and clinical management. So please bear this in mind and restrict your questions to the session on research and clinical trials. So we start the session with Dr. Mark Miravitlis. Mark is a respiratory physician who works as a consultant and senior researcher at the Vall d'Hebron University Hospital and Research Institute in Barcelona, Spain. He's authored a vast output of publications in COPD and Alpha-1 antitrypsin deficiency and has served as chair of the Respiratory Infections Group of the European Respiratory Society and as their guidelines director, and is currently one of the co-chairs of EARCO, having been instrumental in establishing this very successful collaboration of researchers and clinicians across and now even beyond Europe. So he will be speaking on the EARCO registry. Thank you very much, Mark.
Thank you. I would like to share my presentation. Yes. Here we are. Great. I guess everything works. So thank you very much, David. It is a pleasure to be today with you to share our experience with the EARCO registry over the last years. Well, as you mentioned, David, this is an initiative, initially an initiative from the ERS, and the idea of establishing this registry came after we developed the European Respiratory Society statement on diagnosis of treatment of Alpha-1. It was agreed and it was recognized that the systematic collection of data would give us important information about the natural history of the disease in patients with Alpha-1. And it will enhance our knowledge about the evolution and the optimal management. Recognizing that, the next step would be to try to establish such a registry, such a systematic data collection. And for that, the ERS has one initiative, which is the CRC, the Clinical Research Collaboration. And we applied for a CRC on Alpha-1, and it was granted by the ERS, and they provide us with some modest initial funding to start developing our registry. And this was the structure of the initial structure of EARCO. Again, this was an initiative of the ERS together with European Lung Foundation, and the main objective and the main project was the EARCO registry. But it is not the only one. There are also some research projects attached to it. Initially, we had only 3, but as you will see later, we have more than 25 ongoing research projects, or some of them already finished, derived from EARCO. We also wanted to establish collaboration with industry partners, with the national registries, and with the European Reference Network for Rare Diseases. This is our initial meeting in 2018 at the airport in Frankfurt, when we established the basis of what was going to be the registry. And in order to start our research, we wanted to know first what were the unmet needs, both for researchers, but also for patients and caregivers. And it is interesting that patients and caregivers across Europe recognize that improving knowledge of Alpha-1, in particular among general practitioners, ranked the first unmet need. But the second was the access to specialized centers for the patients with Alpha-1. And there was a series of other unmet needs that we have tried to address with our research projects. But going back to the registry, I must say that it has been very successful. Initially, we had to face with the pandemic, so you can see the very low recruitment over the first months, obviously. But then after that, the recruitment has been increasing steadily, even without yet a plateau. So it's still increasing. But even more importantly, we are following the patients or the participants very closely with a lot of follow-up 1 visits, also follow-up 2, 3, and we are starting follow-up 4 for those patients who were recruited at the beginning in 2020. So you can see here the numbers, and I want to take this opportunity to give a special thanks to all the participants, patients, relatives, carriers who accepted to participate and generously gave permission to share the data, the clinical data for our research. I think this is extremely important, and we are very grateful for that. Here you can see the participation by country. Still Spain and then the UK are the most countries with most participants, also followed by Sweden, Portugal, France, and some of the countries that joined later the registry. If you want to have more information about EARCO in general, this is our website where you will find quite complete information about our activities. Interestingly, you will see all the affiliated centers or reference centers in the different countries that participate in EARCO. And in summary, you can see here the updated list of participating centers by country and also the most recruiting centers. You can see that there are very large centers, for example, in Vigo in Spain with more than 300 participants, in Sweden, in Birmingham, in Santiago again, this is our hospital in Barcelona, 145. So a series of centers across Europe and beyond Europe, because you can see how we have also Toronto here in Canada, and we also have centers in Latin America, and we are having negotiations with Australia and New Zealand as well. So we have more than 100 centers in 25 countries. Regarding the research projects, I show you how initially, in addition to the registry, we started with 3 research projects. Currently, we have a lot. We have, I see in my screen, yeah, we have 28 research projects. All of them are briefly described in our website and the status of these research projects. I want to just give you an overview of some of the research projects and some of the results we have obtained. So there was the commitment that we will present the first characteristics of the participants in the registry when we reach the first 1,000 patients included. And this is what we published by the end of 2022. And I don't want to go into details, but just to show you the type of data that we are collecting. So we have quite complete information about lung function. We include data from subjects who are at homozygosis, but not only, we also include other deficient variants. We have SC, a unique group of SS, and also some rare alleles. It is very important, and this was also requested by the patients' representatives in EARCO, that we collect patient-reported outcomes. We wanted to have an overview of how the disease impacts in the quality of life of the patients. So we collect the CAT, which is specific for respiratory symptoms, but we also collect generic instruments. We use generic instruments such as the EQ5D for generic quality of life and the visual analog scale in quality of life. And you can see the scores of the participants according to the different genotypes. Interestingly enough, we wanted to compare patients in the north and the south of Europe. In fact, when we divided our CC population between these 2 areas, the lung function was very similar. But in contrast, the CAT scores were significantly different. You can see in the northern countries a mean of 16, but in the southern countries, a mean of only 9. So that means a much better, significantly better quality of life in patients in the southern countries. And also in this case, half of them were receiving augmentation therapy versus only 4 in northern countries. So it is interesting to see how the disease may impact. There are other aspects that may impact in the quality of life of our patients. And well, this is not unique for Alpha-1. I mean, for chronic respiratory patients, it is well known that milder climates improve their quality of life. So this is why some patients from Nordic European countries, if they can, they try to move to other areas with milder climates that they're not so cold. Another interesting investigation was regarding other respiratory diseases, such as bronchiectasis. And one of the main findings is that we found that only 9% of the bronchiectasis patients had bronchiectasis alone without emphysema. In the majority of cases, bronchiectasis were associated with emphysema. And you can see how patients who had bronchiectasis alone, they have a well-preserved diffusion capacity compared with patients who had also emphysema. So there are different clinical characteristics in these individuals. Another aspect that is quite unique for EARCO is that we could collect information of a significant group of carriers of the SS genotype, which in many clinics, they are not taken into account. They are not included in studies on alpha-1 antitrypsin deficiency. We could see that, well, as expected, homozygous CC had a higher prevalence of emphysema compared to SC and SS. But in contrast, the prevalence of bronchiectasis or asthma were similar between these 3 groups. And even in asthma, SS appeared to have a trend towards a higher prevalence of asthma. Regarding lung function, the CC in general had more impaired lung function, but SS have a similar impairment to SC. So therefore, if we consider SC as a risk for developing lung disease or having an impaired lung function, perhaps we need to start to think about SS as having a very similar risk to the SC. Finally, what I wanted to also tell you today is that having EARCO does not mean that the national registries have disappeared. So national registries in some countries are still existing, and in some countries they are integrated into EARCO. And this is the Spanish example. So all the centers, all the investigators, and all the participants in the EARCO who are based in Spain, they constitute the Spanish registry. And there is one Spanish coordinator, and they have free access to all the Spanish data for analysis, because there may be some questions, some research questions that may be unique or different for Spain compared to other countries. And this is an example with Maria Torres, who was the leader of the Spanish registry at that time. We elaborate this analysis looking at the different clinical phenotypes of patients in EARCO in CC or SC genotypes. And also you can see, as expected, patients with the CC genotype have more affected lung function compared to SC. A second example is Portugal. Also all the Portuguese centers constitute, in EARCO, constitute the Portuguese registry. They call it the Portuguese EARCO registry. And they also elaborate with the national data some prediction model for exacerbations in these patients. So just to give you an overview of how it works. And also, EARCO is not only a database. There is also a group of colleagues, a group of specialists, and also patients that can work together in other type of activities. For example, this is a Delphi consensus about assessment and monitoring of lung disease. You know that there is no evidence about how often we need to visit a patient, how often we need to perform lung function tests, or how often a patient needs a CT scan, for example. And since there is no evidence, the best guidance comes from the expert opinion, including also the patient's views. And the final outcome, obviously, for all research are the publications, to make public the results that we were able to obtain thanks to your contribution to the database, to your contribution with your data to the ARCO registry. And so far, we have generated 19 publications of different aspects of the ARCO, of the general database, but also from some of the research projects that have already finished. And this is the summary, and this is my last slide. So the ARCO registry was launched in February 2020. We have 101 recruiting centers in 25 countries with almost 3,400 participants. There are 28 research projects, of which 9 has been finished and published, 12 are ongoing, and 7 are on the design phase. We are also very proud of our young investigators. We initially identified 15 early career members from 9 countries who were joining ARCO and interested in the research. But now in a second wave, let's say, of call for early career members, we reach 30 from 12 countries that join ARCO, and 6 of them are leading research projects. So we are reassuring the future of ARCO and the future of the registry and the research in Alpha-1 in Europe and beyond. And also we have created a community with a partnership with the ELF, also with the collaboration with patient organizations and with pharma industry. And this is the picture of the ERS Congress 2 years ago with a big group. As you can see, the 3 chairs had to stand in the back because there were not enough seats for all of us. So the family grew a lot, and we are still recruiting more centers and more participants to our project. Thank you for your attention.
Thank you very much, Mark. Our next speaker is Dr. Sandra Nestle-Parr. Sandra is the global head of value and access at an American biotechnology company that develops novel therapies for people with rare diseases. She's also a passionate patient advocate who seeks to elevate the voice of people living with rare conditions, not just Alpha-1. She's been on the board of trustees of the Alpha-1 UK support group for over 10 years and has been an advisor to Eurodis on a variety of topics. And Sandra will be speaking on patient participation in clinical trials.
Thank you, David. Thank you very much for the invitation to speak here. It's a great pleasure for me to be here, and particularly on a topic that's so close to my heart, not only in my day job, but also in my patient advocacy work. So before I discuss patient participation in clinical trials, I thought it would be helpful to provide some broader context related to the inclusion of the patient voice. And this schematic illustrates the different stages from discovery of a potential new drug all the way to patients being able to access a new therapy in their country. And the multi-stage process of testing a new therapy in humans before it can be approved by regulatory agencies such as the European Medicines Agency is called clinical development. And it typically includes clinical trial phases 1 to 3. And decision makers are increasingly requiring that the patient voice is incorporated into all stages of this long process, which can take up to 18 years. So it's critical that patients understand this process, how it relates to their involvement across the board, but particularly their involvement in clinical development. So until about 15 or 20 years ago, a marketing authorization or license for new medicine was sufficient for it to be prescribed by doctors and reimbursed by healthcare systems. However, today, the famous 4th hurdle of obtaining reimbursement needs to be crossed in every single country before a new treatment gets into the hands of patients. And before they make a treatment available to patients, decision makers and healthcare budget holders demand to see that a new therapy offers value for money as they see it. But they assess this increasingly more from the perspective of what matters to patients. And this slide highlights many of the opportunities for patient engagement that drug companies and clinicians that are involved in clinical research are now expected to make available to patients to foster a patient-focused drug development process, all the way from clinical research to approval of a new medicine. And I know this is a busy slide. I'm not going to go into any more detail, but my slides will be available on the website after the conference, so you can look at all the details once you've got access to these slides. And similarly, I don't expect you to read this busy slide, but it is an illustration of the sharp increase in policy initiatives and consensus documents and guidelines by regulators and health systems that we have seen in the last few years. And these documents stipulate the requirements and the processes of patient engagement, and also how patient experience information should be collected and used across the drug development, the regulatory and reimbursement approval processes. So the slide is also meant as a resource for you, and the link for relevant publication is included at the bottom. Now, before we talk specifically about clinical trials and patient participation in them, it would be great to get a top-line reading from the audience here on how much you know about clinical trials. So I think the first poll here has been launched. So it'd be great to understand how much you know about clinical trials. Is it nothing? Is it a little bit? Not as much as you would like, a lot or everything. And I would ask that only patients and caregiver attendees answer this question, please. So I think this is live now. So can I ask everybody to make their vote? And I'm not quite sure. At some point, the results, I think, will show up somewhere. Is that right? Oh, here we go. So interesting. So we have a spread here, and not surprisingly, there are not many people who think that they know everything about clinical trials, and I think the majority sit somewhere in the middle that would like to know more. So that's reassuring, because that's what I'm going to be talking about next. I just wanted to check with the organizers. Did everybody see the results of the poll, or did I just see it? Okay.
Yes, we did.
Okay, fantastic. Thank you for confirming. So this slide summarizes the different clinical trial phases that you may have heard of in the context of potential clinical trial participation. And these phases apply to all new medicines and are not specific to trials related to Alpha-1. So starting on the left, before any testing in humans can begin, so-called preclinical studies gather information usually in a laboratory setting about the way a new drug works, the feasibility of using it in humans, and its safety. Phase 1 is the first clinical phase, often also called first-in-man studies, and they typically involve small groups of healthy volunteers to assess the safety of the drug. Phase 2 studies involve slightly larger numbers of people with the condition to be studied, and they assess the safety, the efficacy, and also the optimal dose and side effect profile of the drug. In phase 3 trials, the drug is tested in a large group of people, which is obviously a relative in a rare disease, but larger patient group that have the condition, to compare the new drug, to current therapies or placebo treatment, and to assess its effectiveness and safety and longer-term effects. Once a drug has been made available, has a license, is available in the market, and has been shown to work, phase 4 studies may be conducted, and they examine how well the drug works and how safe it is when it's used more widely outside a clinical trial setting in routine clinical practice. And there are many different ways to design a clinical trial, and examples shown here of very common study designs that you may have come across are studies where no treatment is given to participants as part of the study, seen on the left. And participants are usually followed and observed, and sometimes over long periods of time. And a good example may be a disease registry, such as the YARCO, which Mark has spoken about, or national Alpha-1 registries that some of you may be enrolled in. Observational studies in Alpha-1 could, for example, include patients who are on specific treatments like augmentation therapy, but also patients who are not on any specific treatment. When we talk about clinical studies in phase 3, sometimes also referred to as pivotal or registrational trials, the gold standard method for a clinical trial is considered a randomized clinical trial, or RCT, which is highlighted here in green. So what does randomized and controlled mean in the context of a randomized controlled trial? There are numerous different trial designs within that group, but they all have two key characteristics in common. Controlled means that the new treatment is studied head-to-head with another treatment, which can either be a placebo, so that's a pill or an injection that does not contain the new drug, or another specific treatment for the same condition. Randomized means that trial participants are assigned randomly to the study groups to receive the treatment, including placebo. So a typical example is a parallel group design that you're likely to have come across, where one group of patients receives only treatment A, let's say that's the new treatment to be studied, whilst another group only receives treatment B, and this could be a placebo, for example. A crossover trial design, which is also fairly common, each patient gets both treatments sequentially. So each patient receives the first treatment, and after a washout period, they then move on to receive the second treatment. So they sort of act as their own control in this trial. And to avoid bias with the clinical trial participants or the study investigator, neither of them know which patient receives which treatment whilst the trial is ongoing. And that's also what's referred to as blinding the trial. And there are other trial designs, which I'm not going to go into, like adaptive trial designs, which are becoming more common. So the issue with rare diseases and running clinical trials is that rare diseases pose a number of different challenges for clinical trials. And common challenges that apply to many rare diseases include that rare diseases by nature have small patient populations. They can be complex in disease presentation, and they often impact more than just one organ system. And heterogeneity in how the disease presents in different people could also pose a challenge. And unless a rare disease is included in a newborn screening, for example, they're often diagnosed late or they're misdiagnosed. It can be really difficult to measure the benefit of a new treatment, because in rare diseases, there are often no disease-specific validated outcome measures that regulators want to see. But in addition to that, Alpha-1 has some further challenges that are specific to the disease. So for it being a genetic condition, there's Alpha-1 starts relatively late in life in general, except for maybe childhood liver disease. It's also a slowly progressing disease usually, and that requires long trial durations and potentially large trials to show the benefit of a new therapy. The increasing challenge in Alpha-1 will be that with the increase of companies interested in developing a treatment, although this is great, it leads to competition for participants because there are many clinical trials that may be ongoing simultaneously. But there are also increasing ethical considerations related to running placebo-controlled trials in Alpha-1 because there are approved treatments, and where there are approved treatments is always an important ethical question as to whether a patient should be on a placebo in a clinical trial. So just to be absolutely clear, without patients participating in clinical trials, it would be impossible to develop new effective therapies for Alpha-1 or any other disease, in fact. However, it is critically important to get a really good understanding of the trial, what it would involve for the patient, and to fully consider a number of important aspects before deciding whether or not to participate in a trial in principle, and which specific clinical trial may be suitable for any particular patient or may not be suitable for that matter. So it's obviously important to understand the investigational therapy. So what drug is being studied here and how does it work? What are the potential risks, side effects, discomforts of the administration itself, and what are the potential benefits that one can expect when receiving that drug? But it's also important, and it's not often talked about, that patients should really understand what the longer-term implications are of participating in a particular trial. So if we look at the treatments that are available, are currently being studied in clinical trials in Alpha-1, there's obviously replacement or augmentation therapy. So that's a fairly short-acting therapy. So after a trial, for example, if the patient comes off the therapy doesn't stay in the body for that long. So it doesn't usually pose a problem in the long term. There are RNA-based therapies that are now moving into clinical trials. They are longer-acting therapies. And DNA-based therapies, such as gene therapy, which you will have heard of, they are designed to be irreversible. So it's really important to understand that participation in a trial with a gene therapy, for example, is irreversible, but may also prevent anybody from getting any other treatment in the future or participating in any other clinical trials in the future. It's obviously really important to understand what the clinical trial itself involves. And again, I'm not going to run through this list. It will be available after the conference to you. But just think really carefully about how long will I have to be in the trial or any follow-up observational period? What does it mean in terms of the number and frequency of visits to the study site? What kind of tests will I have to undergo? What happens after the trial? Will I be able to continue on the treatment when the trial is finished, for example? As I already mentioned, it's really important to be clear on whether participation in a trial may potentially exclude you from participating in any other trials in the future, and for how long. When participating in a clinical trial, it's very important to also think how practically in everyday life participation would impact a participant, but also their family. So how much time will you have to potentially take out of work to go to hospital? How often and for how long? There are trials, gene therapy is a good example, where participants might be expected to stay for several days in a clinic and stay there overnight to assess whether or not they're even eligible to participate in the trial. So these are all important considerations. There's the cost consideration. So might you have to take time off work? Might you need additional childcare that you have to pay for? What about transport to the clinical trial site, or any costs related to that? Is that covered? And who can you talk to in order to get in order to get a really good understanding about all of these questions. What's also really important is to discuss what are your choices before you consider going into a clinical trial. So there are countries where there are already treatments available for Alpha-1. There may also be other clinical trials that may be available, and not every clinical trial may resonate with you equally well. So it's important to understand what other clinical trials are currently available that may be an option for me, or are likely to become available in the near future. It's also a really important question to be clear about how you might feel if, whilst you're on the trial, a new treatment may become available routinely in your country, and you could access this to the national health for your national health system, but you're still on the trial. So once you commit to a trial, it's really important to be clear about these questions, because if you discontinue from a trial, it could really put the success of the trial at risk. So moving on to another poll question, I'm sure all of you will have heard of informed consent. And the question for you here is, if a patient signs the study consent form, this means that they have given their informed consent to participate in the trial. Is this statement true or is it false? What do you think? So we're just waiting here for the results to come in. So I think we can show the results now if they're available, please. So interesting. The majority think that this statement is true. Well, I admit it was a little bit of a trick question because it's actually not true. So informed consent, which is one of the founding principles of research ethics, so unlike contrary to common belief, it is not just the process of signing a consent form. It's often mistaken as that, but it's really important to understand what informed consent really involves. So it involves the responsibility for the sponsor or the investigator to provide all the relevant information in an adequate form to potential participants, but also to facilitate that potential participants understand that information. So it needs to be delivered to them in a way that they understand, and the investigator needs to make sure that the patient or potential participant has understood all the information. It's really critical that any consent has to be given freely of your own will, and that this process must, the process of agreeing, must minimize the possibility of coercion or undue influence. So you should never participate in a trial because you feel your doctor wants you to, or you're expected to because you already spent a few days in the hospital being screened for a trial. You've got to be absolutely clear that this is your free decision. And you should also discuss it with your family, with friends, with your GP, with the doctor who looks after you routinely. And the study investigator is responsible for continuing to provide information as the clinical trial proceeds. And importantly, it is important to know that consent to participate in a trial can be withdrawn at any point before you join the trial or once you're in a trial. So in the interest of time, I'm not going to cover this in too much detail. I said most of these things, but it's your responsibility to really be clear about all of those questions that are relevant in terms of participation in a trial. Be absolutely clear that you understand the information. Ask as many questions as you feel you need to fully understand it and feel comfortable. And also understand what happens if you decide to discontinue your participation in the trial. So what will be the consequences and what happens afterwards. So in summary, there are lots of things to consider before you make a final decision to participate in a clinical trial. Discuss it broadly with your family, your friends, your doctor. Be clear about why you want to participate in the trial. Is it because you think you may get access to treatment that you otherwise don't have? Is it because you want to help other people with this disease in the future? And then you need to be clear whether the commitment you have to make is sufficient for you to see it through. You also need to be clear on how you might feel about potentially not getting the active drug, but in a placebo-controlled trial, you could get the placebo. So how might you feel about remaining in the trial if you feel that whatever treatment you're given may not help you? And finally, it's really important that if you're comfortable with the trial site, with the institution that's running the trial, and the people you're in touch with on a day-to-day basis as it relates to your trial. So just one last point, why it is so important for patients to be absolutely clear about all of these issues before they agree to participate in the trial. You have to feel comfortable and confident that you will be able to commit for the duration of the study in whatever is involved in the study. Because if you don't, then it really compromises the success of the trial. And every new investigational treatment should have a fair chance of being adequately assessed. And that's not possible if a lot of patients discontinue on the trial whilst they're on it, which doesn't mean to say that you cannot discontinue. You absolutely can, but to minimize that possibility, you should be absolutely clear about all of these points before you join the trial. And with that, I'd like to just finish with our last poll, if we could please pull that up. So after we've heard, how confident are you that you know what questions you would need to ask before deciding whether you would participate in a clinical trial? Are you not confident at all, slightly confident, fairly confident, or fully confident? So please vote. And again, I would only ask patients and caregivers to answer this question. And if you could have the results, please. Great, so that's really encouraging. So it looks like the majority of participants here, 60%, are fairly confident. And I think this was one of the objectives of this talk, to give you that confidence. So with that, thank you very much. And I'll hand it back over to David.
Thank you very much, Sandra. We move rapidly on now to our final speaker of the session, who is Professor Alice Turner. Alice is a respiratory physician at the University of Birmingham, UK. I've known her since she was a junior doctor and trainee in respiratory medicine. And when she started her research, which initially focused on the genetics of Alpha-1, she continues to be very active in AATD research and heads the Birmingham Alpha-1 service, which has evolved from the ADAPT program. Alice will be speaking on current Alpha-1 antitrypsin deficiency research.
Thank you very much. So I've chosen to try and give a little bit of an overview about types of research, understanding that some of it has been presented already by Mark and Sandra, and hopefully introduce people that are unfamiliar with the different types of research to how they differ from one another. So we'll go through what types of research there are. We'll briefly cover some examples of each type of research in Alpha-1. And during the time that I'm talking about them, I hope that I will be able to bring across how each kind of research can benefit patients. And then very briefly at the end, I'll cover some ways to get involved. I know that in the Q&A and in the chat, people have been asking about ways to get involved in trials, and some colleagues have been answering those questions as we've gone along, although the processes may differ in different countries about how you'll find information. So in brief, what kinds of research are there? I've divided it into 4 types that I'm going to talk about and 1 type that I won't talk about today. So basic research essentially will cover things like molecular types of structures, how molecules affect processes within cells, and this is stuff that will be conducted in a laboratory. Epidemiology and database studies, you've heard a lot about the principles of that from Mark when he was talking about EARCO. And epidemiology and database studies are the kinds of things that will help us to understand how common a disease is, how it varies between different patients. And then that leads on to us understanding what we might need to do next to help those patients to feel better and to treat them appropriately. So for those 2 types of research, I've illustrated that here with a molecule. In the basic diagram, what I've shown you is a picture of the alpha-1 antitrypsin molecule. And on it, I've marked where the reactive loop of alpha-1 is and the mutation that can occur within it that then changes the way that it functions in the ZZ genotype. And here you can see an epidemiological study demonstrating the number of known ZZ alpha-1 patients in different countries of the world. And you'll note that large tracts of the world have got white coloured in, and that's because we don't have good data in those countries at the moment. I've illustrated qualitative research with a picture of people talking to one another. Qualitative research is research where we will gain information from getting patients to tell, or indeed healthcare providers, to tell their stories to a researcher. And we will use that when we want to really get in depth into some data that maybe we don't understand where to start yet. And so we need to get a broad picture from the people that we're talking to. Clinical trials, you've heard a lot about from Sandra, and I'm not going to talk to you in detail about that, except to show you the types of trials that are currently ongoing in Alpha-1, so that you've got an idea of the big picture going on. And then finally, the type of research I'm not going to talk about today. There is a wide variety of animal-based research. I'm not going to talk about that today because I wanted the focus here to be about the types of research that you as a patient might be approached for. And animal research is not something that you will be approached for personally, so I felt it was less important to dwell on that today. But if questions do arise, I am more than happy to take them. So let's go into each one of those now in a little bit more detail. So basic research is there because we need to understand the pathways that lead to disease. So from a genetic basis to a protein basis, and then onto the cells and the organs that, that we understand so that we can diagnose and treat disease. And basic research can be theoretical. So, for example, we might look at genes and then use modeling programs like one called AlphaFold that has been generated to help us learn theoretically how a protein would change if we changed the Alpha-1 gene. And this kind of theoretical research has led us on to cellular-based research that has led us on to disease mechanisms and also to treatments. So here what I've done is illustrate some understanding of the nucleus of the cell, which is where our DNA is held. And here I've illustrated the SERPIN-A1 gene, which is where the Z mutation occurs. By understanding that this DNA was here and what the mutation was, we've been able to develop studies of gene addition and gene editing over the years. Understanding that DNA makes RNA has allowed us to develop RNA editing studies. And understanding that the mutated protein in the Z, in patients carrying the Z allele, accumulates within the endoplasmic reticulum, which is part of a liver cell, and then causes those cells to be stressed, has helped us to then lead on to other forms of liver-directed therapies. Another example of basic research is some work that's been conducted by colleagues in Birmingham over recent years. And what they've done is try to make a model of how Alpha-1 would work within the lung. And to do this, what they've done is take cells from patients who've given consent to have their blood taken for research, and they've put out different concentrations of Alpha-1 with that to mimic the different types of Alpha-1 that we see from a genetic level. They've then challenged those cells with something that would potentially mimic inflammation. They've then collected ingredients from those cellular experiments, stored them in a freezer, and measured some things that we know are important to demonstrating the process of protease imbalance, which is ultimately what damages the lung. And what they found when they finished these results was that if they measured one of these downstream biomarkers known as Alpha-541, that we could see the levels of that go down when we had more Alpha-1 in the cellular experiments. That suggested that Alpha-1 antitrypsin was maybe damping down the effects of this within the system. And in fact, we can see that in patients who are deficient in Alpha-1, when we later went on to measure this in patient blood, that we saw different concentrations with people who got the ZZ genotype compared to people with COPD who had the MM genotype. So in effect, what this is telling us is that we now have a cellular model where we could test things that might be relevant to a ZZ Alpha-1 patient in a cellular model so that we can start to develop new drugs before we get to the stage where we're ready to think about them in clinical trials. And there are many different types of this research that will happen, and we'll talk about how you could get involved in that kind of research at the very end. Epidemiology and databases are probably one of the most common forms of research that patients will be approached for, mainly because many large numbers of patients are needed in these types of studies in order to help understand things. And this is because we need to understand patterns of disease to help us to develop both treatments and services. Much of the epidemiological research that you've heard about so far involves specific data collected to research databases like EARCO. But epidemiological database research can also use the routine care record, in which case the models for consent are slightly different and will involve much higher levels of regulation if patient records are going to be used without consent. And that process is dealt with by governments in different structures countrywide. The levels of detail and accuracy in these sorts of studies will vary according to the type of data being examined. So with EARCO, we collect detailed data and patient consent is taken so that we can make sure that we don't have too many gaps within the system. But if we were to use a GP record, for example, that had been collected routinely for some other reason, but there would potentially be lots of missing data and the accuracy might be less good. Nevertheless, those sorts of research have validity and they can be helpful in answering different kinds of research questions. Just to give you an example of some epidemiology research outside of EARCO, because you've heard about that from Mark already, this is an example from several different studies that have combined to help us understand liver disease in Alpha-1 antitrypsin deficiency. So liver disease, as I showed you in the basic research slide, can cause stress to liver cells. And ultimately that stress to liver cells can lead to the development of fibrosis. We talk about fibrosis in different ways, but we measure it on a scan, and this can help us to give it a number. F2 fibrosis is a sort of medium level of fibrosis to the liver. And we would expect from epidemiology that we would see that in around 30% of patients with the ZZ genotype in adulthood. However, if you're in a hepatology clinic, there will be many more patients with that because, of course, we want our patients with liver disease to be seen by liver specialists. Liver disease has been shown in these studies, which have included very large numbers of patients, 482,000 participants in the UK Biobank, of whom 141 had the ZZ genotype, to associate with being male, being older, being overweight, and having diabetes. In smaller studies, such as the ones here documented by Clark in the States, they found that if you biopsy the liver, the type of scan that gives you this number agrees very closely. And an AUROC is a measurement that we would use to tell us how well those numbers adhere to one another, and whether one test could be used to tell us about the other. So that gives us confidence that we can scan the liver without needing to do a biopsy and still have accurate results about how the liver is affected. Qualitative research analyzes conversations, but like any kinds of research, we still have to demonstrate that our results are there and what it is that we're finding. So the results and the way that we demonstrate those results is to provide quotes from what people have said. So you can't just tell a story without showing the data. And the data in this case is the quotes. So a typical qualitative study will involve an interview, and then two separate people will independently look at those interviews, usually in a written form after it's been transcribed, and they will assign codes to help know what kinds of themes were being covered by people. And each of those themes will then need to be proven with a quote. A couple of examples of qualitative research in alpha-1 antitrypsin deficiency include studies of the effect of the disease being diagnosed within a family and the effect of having a diagnosis on subsequent relationships. These studies are a little bit older. This type of research has been less widely conducted with an alpha-1, or at least less widely published, but is still very valuable. So in the first study, a family who had had liver disease diagnosed in a sibling in childhood, and that sibling had been very unwell with that disease and ultimately had unfortunately died of their liver disease. Their sister said, "I can remember just riding her tricycle in the meadow. It would phase into her being a hospital in my dream. And the hospital would get on fire and flames would be all over the place. I remember trying to go get her, trying to go find her, and they wouldn't let me go." So she's illustrating there the impact emotionally that her sibling being unwell had on her. And this led on to recommendations for family testing and counselling after this work was done. Other researchers looked about the impact on relationships. And so this then led on to recommendations for counselling and for public health to help address the concepts of genetic diseases and help people to bring them up when they start talking to others. There could be pressure put onto a relationship for thinking about children, for example, if you don't know how to bring that up with a potential partner in the future. And this is illustrated by the quote you can see on the right. Sandra's given a really good overview already of clinical trials. So I just wanted to give you a very quick overview of trials that are ongoing at the moment that have potential to help the lung in the long run. Early phase trials, as Sandra outlined, are mainly around safety, and we can't be sure at these stages how well if those drugs will benefit patients. As we go through later phases from 1 through 2 to 3, we may be gaining greater degrees of confidence that those drugs will help patients, but we won't know until we've finished those trials. There are a variety of DNA editing studies that act within the DNA, RNA editing, inhaled alpha-1 studies, and also studies of synthetic augmentation that are ongoing at the moment. Every trial will have what's called a primary outcome, and these are the ones that are being measured in some of these trials that are designed primarily at thinking about lung disease patients in the first instance. But the DNA and RNA editing studies will also potentially help liver disease. There are also some trials that are focusing very much onto liver outcomes. And so here you can see the DNA and RNA editing studies, but there is also an RNA interference study that is at quite an advanced stage of development that is looking at a liver biopsy as its main outcome. There are also a suite of folding correctors that act within the cellular mechanism that you saw in the basic science slide earlier, These may also help the lung in the long run, but in the first instance, the trial companies thought about using liver biopsies as the main thing that they would be looking at to tell them if it was helpful. At the moment, these trials are on pause, but the company hope to be coming back to this space in the near future. So how can you get involved? The consent process has been described in detail by Sandra already, and researchers must at all times adhere to good clinical practice, which protects patients participating in research. You could be approached for consent for basic science studies, which you often use blood or sputum samples or DNA. Database research may also involve consent and could mean additional questions compared to normal medical care and may mean repeat visits as well. Qualitative research also would always involve consent and has detailed interviews, but usually these would only occur once. That's where I wanted to end it, and I hope that I've left plenty of time for questions.
That's lovely. Thank you very much, Alice. We do have quite a few questions. Before you disappear, Alice, I think what I'll do is I'll ask a question that's been put through that probably is best suited to your area of expertise, and that is someone has asked the following question: what would be the good animal model for alpha-1 antitrypsin deficiency? The recent clinical data have shown that levels obtained in mouse PIZ model do not translate well into the human.
So that's a really great question. The Alpha-1 Foundation are running a specific session in the near future by researchers that are focusing on animal models to talk about the best ones that there might be. For lung disease at the moment, the leading model is thought to be a ferret model. And this is because ferrets, unlike mice, develop much more similar features of lung disease compared to humans. So, for example, mice don't typically develop coughing up of phlegm quite so much, although they will develop emphysema, whereas ferrets will develop both the bronchitis element that typically leads to sputum, as well as the emphysema element. So in summary, I think the ferret model for the lung is probably the best one. Liver disease models, I think that Pavel, who is talking later, would probably be a lot more better qualified to comment upon. But the mice have been challenging because they have lots of different types of alpha-1 within their system, and that makes them harder to study for the liver.
Wonderful, thank you very much. And do you see that there may be a cure for alpha-1 on the horizon?
So I remember when I was doing my PhD, which is equally many years ago now, making me feel old. And Rob Stockley, who was my supervisor at the time, said, there will be a cure in my working lifetime. He's no longer seeing patients, so there wasn't one during that time. But I do strongly believe that there will be a cure within my lifetime that I may see, because there are so many different options coming through in both clinical trials and in terms of our understanding of basic mechanisms that I have to be hopeful that at least one of those will come to fruition.
Good. Well, I hope I'm around to witness that, hold you to account. Thank you very much. So I have a question for Mark. So this is a very quick question. One is relating to the EARCO website having a consent form for patients to view, which is only in English. I wonder if you could just comment on that and also confirm that in each institute participating in the registry that they do have the consent form in their native language.
Yes, that's correct. No, there are consent forms in every language. And in fact, they are in the website for download, but this is a model. And I think they have two versions, one in English and one in Spanish in the website. I'm not completely sure, but these are only models that need to be translated and adapted to the local language and the local legislation, because not in all countries, the forms are exactly the same or include exactly the same information. But in any case, the patients themselves should not sign this document. If they want to participate in the registry, what they need to do is to check if there is any center near where they live, and then go to that center or through their attending physician, contact that center and then request the participation in that center, because the patients don't register themselves. All the registry goes through the investigators in the reference centers.
And could you clarify which genotypes are being included? I think someone may have, I think Christian may have written a response to that question, but I think it's quite an important one to emphasize.
I think it's easier to say which are not included. Those who are not included are those who include at least one normal M. Obviously, if you have two M's, you are not deficient, but if you have one M, irrespective of what the other allele is, you don't qualify for to be included in the registry. So you need to have two deficient alleles.
And could you also briefly outline to patients who may be interested in joining the registry what that actually means in terms of their commitment, the time, and how things might differ from what they would normally experience if they were just attending a clinic for their treatment?
Yeah, not much really, because the registry is an observational study. Observational means that we don't intervene. We only observe what happens in real life. Having said that, we request PROs, questionnaires, and some of these questionnaires may not be used at every center at every visit. But this is a minimal effort in terms of dedication or time. So we ask for one baseline visit and one follow-up visit every year. So we only request one clinical visit per year. And with this, the normal follow-up, which is lung function, a blood test, and the PROs. I mean, for the majority of participants, if they have some degree of lung disease, it will not mean anything extra. It's different for individuals who are non-index cases, who may have healthy lungs, and who otherwise may not require complete lung function test every year. And by participating in the registry, we ask them, please, to have complete lung function and a blood analysis every year. And in these cases, yeah, you can say that perhaps these people that are not yet affected may require some more testing. But in any case, we believe that subjects that have these two abnormal alleles, it's good to be attended in a reference center and have at least one yearly visit with some diagnostic tests.
Okay, so you mentioned how it was easier to list those who are not eligible. So you did say that you didn't view the MZ as being deficient. I wondered, for the sake of clarity, because I suspect that there are quite a few MZs that may be participants in this webinar who may be under the impression that they have Alpha-1 antitrypsin deficiency and may therefore take some of the messages that are discussed around Alpha-1 deficiency home with them, thinking that it applies to them. I wondered if you could briefly just talk about your perception of how much of a risk the MZ genotype constitutes.
Mm-hmm. This has been a matter of debate, but in the studies we have suggest that the MZ do not have an increased risk of lung disease if they don't smoke. But if they smoke, they have an increased risk compared with the MM normal population. So the best treatment for them is to quit smoking if they smoke or never start smoking. We know that without a significant exposure to smoking or other inhaled risk factors, they will not have an increased risk of lung disease. And there are no other therapies for them. That's why we don't include it in the ARCO registry. Perhaps Pavel later on will talk about liver disease, which is different. Having a single C allele has an increased risk of liver disease compared to an MM and less than a CC, and therefore talking about the liver, there's a different issue. But for lung health, for the lung disease, an MZ does not have an increased risk if it's not exposed to smoking.
Thank you very much. Sandra, I wondered if I could ask you a bit of a tricky question, but I'm sure that you're the best person to answer this. And that is, given the difficulties of undertaking clinical trials in patients with rare diseases, I wondered if you could talk about what the regulators are trying to do in order to make allowances for these difficulties.
Yeah, you're referring to the challenges, some of which I talked about in doing clinical research, particularly in small patient populations and rare diseases. So I very briefly mentioned trial designs that may be accepted by regulators in order to address that, such as adaptive trial designs. Now, I didn't go into that, but these are essentially trial designs that can change over the course of a clinical trial in response to what the investigators are seeing. And this is often done to make up for the fact that you may not be able to include a lot of patients in your clinical trial. So regulators have some flexibility around what they're expected to see, but it's also fair to say that in the last maybe 10 years or so that they have become a little tougher on rare diseases because they expect companies that develop rare diseases to really do their homework before they embark on a clinical trial. And there's some key parameters that regulators still want to see, particularly relating to the clinical outcomes that are being measured. You know, they should be validated, ideally in the particular disease that's being studied, but also in terms of the safety. I mean, safety is absolutely paramount. And as we talk about gene therapy, for example, you know, patients on gene therapy may be requested in a gene therapy trial, they may be requested by regulators to have a follow-up to 15 or 20 years. So regulators try and do what they can in order to facilitate new treatments becoming available to patients. But it is tricky, and this is why it's important for drug companies to really think very carefully how they design the trial in order to maximize the chances for it to be successful, but also for patients to choose well.
Wonderful. And I wondered if you could also talk a little bit more about how patients can learn more about clinical trials to get independent advice about specific trials. So I have on occasions discussed clinical trials with my patients and have suggested that they get independent advice and talk about it with their family and other doctors, some of them with doctors in the family or with their GP. And quite often the response that I get back is, well, I spoke to my GP and he really doesn't feel that he's qualified to give any advice. Is there somewhere which is independent, central, where you can get more structured advice for patients on particular clinical trials?
Well, I mean, the particular clinical trials is obviously usually the developers that provide and are legally responsible for providing information for patients, which is also designed in a patient-centric language. But of course, you're right. You know, somebody, the company wants to do the trial or investigator, they may be not entirely independent to advise a patient because obviously they want to run the trial, which is understandable. So there are many resources, and some of those are included actually in my slides. I've got a few links, so once they become available, people can take a look at that. But you know, going to your orders, going to NORD, which is the North American equivalent of your orders, going to the FDA, the EMA, these are good. These organizations provide good information, but they can also act as a signpost to direct patients to additional resources. Now, trial-specific information is difficult to get it anywhere else than from the developers of the trial or the investigators that are conducting the trial. I think that's fair to say. It might also be, I mean, a lot of patients communicate social media, etc., and they exchange information about their experience of participation in a particular trial. So there are avenues, but you know, I agree with you. I think patients need to be advised to also seek information independently in order to make up their mind.
Great. And then there is a question which is addressed to you. I don't know whether or not you want to type an answer to that. I can't quite follow some of it because it's very technical, but it's about the use of artificial intelligence to mimic control groups, and it refers to some specific projects that I'm not familiar with, and also relating to toxicity safety testing and non-clinical studies. So that may be something that you could type an answer to, because we are running out of time. And then lastly, there's a question about PROs, which ones the EARCO is measuring, and I wondered if perhaps Alice could answer that.
So there are measures of symptoms that we look at. So, for example, the CAT score, and also that help us with health economics, such as EQ5D, and also some measures of physical activity as well. So those will all be presented after the consent process to patients.
Wonderful. So just to finish off, I think we've fairly comprehensively covered the issues relating to Alpha-1 research at the present time, both from the perspective of active researchers, the value of joining a registry, not just in terms of improving the understanding of the disease, but also potentially acting as a gateway for patients to enter research studies, and also to look at the perspective for future research as outlined by Alice. So I'd like to thank all of our speakers once more and draw this session to a close. Thank you very much.